Cool. Okay, so I promised to talk a little bit about focused ultrasound, but I'm not going to. So why are you interested in it? Suppose that you have a couple of objects in a brain that appear to be correlated with each other because their fluctuations look like each other and have some correlation. If you're interested in testing whether that's true or not, you could zap one or the other and look for changes in that correlation, and that might tell you something about the connection between them. Often there are hidden regions of the brain, and you could test that as well to see whether, in fact, there was a connection between them that was responsible for the correlation. Now, if you do such, you will have a more causal relationship that might be telling for you, and it's much more powerful than a simple correlation. So one such technology is transcranial magnetic stimulation, in which you put a coil next to the subject, and you put a large current in that coil, maybe 4,000 or 5,000 amps for a few hundred microseconds. You generate a change in the magnetic field that generates an electric field. The electric field can modulate the local electrical potentials near membranes, and thereby change the synaptic or the firing activity within a localized region relative to where the magnetic field was induced. So that's one way, and here's an example of a study done at Stanford in which one was interested in looking at changes between the central executive network, the salience network, and possibly influencing the default mode network, 3 separate network systems. So targeting either with positive or negative, that is excitatory versus inhibitory TMS on the dorsolateral areas, did it affect the default mode network? And the answer is yes. On the left, you see that excitatory TMS to central executive did decrease the DMN, and on the right, showing an increase in the DMN from connection distant to the DMN nodes. And there's been many studies of the interactions between TDCS and various resting state networks. Here's one, here's another, here's a third, and we'll see that many of these conclusions are a little bit disparate with each other, but many people have started looking at the influence of transcranial DC stimulation and resting state or even task activation. So transcranial electric stimulation, whether it's DC or AC, uses an electrode placed on the brain and somewhere else, or perhaps another region in the brain. Here's a more modern look at what these systems might look like, and it turns out that with TDCS, you put a constant current on for some period of time, and then you look for a change in local activity. With AC stimulation, TACS, the alternating currents can perhaps interact with endogenous oscillations going on, that is, coherent activity within the brain. So transcranial electric stimulation, which includes both of those, one has to worry when you put such a system in the brain, in the fMRI, because of induced RF currents that could come along the electrodes. So one uses resistors and inductors and so on to make sure that you don't burn up the person, because that's not good. You can also worry about inducing artifacts in the images, so that you have to filter things and so on. And you also have to worry about high SAR, a large deposition of RF, when you're doing these kinds of things in a magnet. But those can be performed. So here's an example using a very simple sensory motor cortex kind of a task using flickering checkerboard as well as passive activation of the fingers using little plungers. So the alternating activity there is a 15-second on-off signal in the visual cortex. The red line shows where stimulus was applied for about 6 minutes, and in the red line, there's no pointer here, so the red line shows the envelope of the activity in the visual cortex, which doesn't change when the TDCS is turned on. But here in the motor cortex, which is where the electrodes were placed, one on the left and right, and we're showing the difference in activity in the left versus the right, you see an increase in the overall activity in the motor cortex that decays away after you turn it off. Time constants are in the order of 9 or 10 minutes or so. Another example of that is a psychophysical interaction where we turn on the TDCS during, again, a constant motor stimulation. Here's the main effect where you see essentially activation in the motor cortex, and the interaction, psychophysical interaction, shows activation in changes in the thalamus and in the basal ganglia, which you expect. And yet another example looking at functional connectivity in a resting state brain between the two blue regions. You see a typical region of interest. We put on TDCS then, and we see again a change in the functional connectivity between those that has again this time constant of on the order of 9 or 10 minutes. One other example, looking at impulse control in a go, no-go task. So we have a 20-minute TDCS stimulation, and during the last 9 minutes, we do a go, no-go task, and we repeat that then, first with a sham that is turning off the current after it's turned on briefly at the beginning and then turning it back on at the end. Then we do a go, no-go, and then we do another one with the real TDCS, and then we look for activation changes. So what you see in the top is the sham, which is compared with the baseline, and basically no signal change. In the second line, we see a change in the inferior frontal gyrus and DLPFC, which is especially what we expect to see with enhanced go, no-go impulse control. And we can look at another contrast and we see the same thing. Now, switching to AC stimulation, it's different because there's no anodal cathodal effects. Rather, the alternating current can engender changes between the ongoing endogenous synchronous activity. And so you basically may be able to modulate the ongoing AC signals that one sees with electrophysiology in the brain. So here's one example. And they used 2 currents, not just one. The bottom and the top electrodes have a common electrode on the in between them, and you put, in this case, 6 hertz AC, either in phase or out of phase between the 2 nodes, the parietal lobe and the frontal lobe. And what they found, and so they did this during a task, and the task was a working memory task, an in-back, which is either 1 or 2. And so they turned on the TACS either during the task with in phase, 0 degrees, or out of phase, and sometimes during the resting state or the baseline region. What they found behaviorally was that in-phase TACS improved the reaction time. So if you look on the right, you see a decreased reaction time during the 0 degrees, but not with the 180 that is out of phase. So it had no effect. And it only affected really the 2-back task, not the 1-back, and that corresponds to a number of other studies in which stimulation tends to change only when you're doing a more difficult task. In the BOLD study, the BOLD concomitantly acquired data demonstrated a nice relationship between the reaction time being faster in those regions of the brain that had a larger BOLD response. Sorry. So we also saw that an increase in the working memory regions was engendered by the in-phase activity. If you did out-of-phase, it affected the default mode network negatively. So that's also an interesting finding. Here's a second study in which the study investigators used the individual alpha frequency of the individual, which is around 10 hertz, as a carrier, which was then modulated at 1 hertz. And so either using in-phase or out-of-phase modulatory activity, and they looked at various conditions within using TACS, again, 2 separate current sources over left and right motor cortex, either in phase or out of phase. And what they found, if you see on the left in the blue curves, an increase in activity relative to baseline using in phase, but not really much of a change when you used out of phase on the right and on the right. What they found indeed was a change in the activity within the motor cortex, but not really in any other network. And yet here's a third study, again using 2 different currents. And if you see on the left, they had one circuit on the left and another circuit on the right. And what they used was currents that were at a frequency very high compared to that of the spiking activity or any kind of electrical activity, so high, like 1 or 2 kilohertz, that they could not in themselves induce any change in the electrical activity of the brain. But they used one condition in which the frequencies were almost the same and only differed by about 10 hertz. And so what you see in the middle plot at the top is when the 2 currents are at 2 kilohertz and 2.01 kilohertz, you see a 10 hertz envelope of temporal interference between the 2 when the currents are comparable. And that plot below it shows what happens when one of them is very weak compared to the other. So the top plot is consistent with the plot on the right, which shows a sweet spot of 10 hertz activity midway between the 2 electrodes, and if you go to one side or the other, that activity goes away. So by using a temporal interference, this Boyden group demonstrated a steerability, because by changing the current ratio between the left and right, or changing where they are, you could steer the sweet spot around and thereby have a way of targeting. In this case, they're interested in deep brain regions. So these are simulations that show by changing either the location or the amplitude of the currents, you could steer in some measure only the low frequency component of the signals. Well, what they found in this set of plots, the left one is a 10 hertz activation or frequency of the stimulation. You get a large change in electrophysiology relative to baseline. If you use either 1 kilohertz or 2 kilohertz with the 2 frequencies separated by 10 hertz, again you get a large change in spiking activity relative to the baseline plots on the right with either 1 kilohertz or 2 kilohertz, showing again that the presence of the interference between the 2 frequencies was needed in order to change things. And so they showed with CFOS activity that, and it's hard to see in these plots, but basically there are 3 regions of interest. If you look on the left at the top, 3 regions of interest in the rat brain close to the electrodes near the cortical surface, and a 4th region of interest below it in the hippocampus. And you see the 3 or the 4 regions of interest are shown to the right. In the top line, it's 10 hertz activity in both electrodes. And you see an activation in the hippocampus, but not in the regions close to the electrode, whereas with the 2 kilohertz in the bottom one, you see activation again in the deep region of the brain, but not close to the electrodes. And if you just use 2 kilohertz on both in the center, you see no activity. So it was, again, the requirement of having a low frequency interference that allowed a spatial representation deep within the brain. And that's shown in these plots as well, which I won't go over. Now, it's interesting that in those cases, if you just look at what's the content of the frequencies present, if you do a sum of 2 sine waves, in this case a toy example with 10 hertz and 11 hertz, you get this temporal interference. And this is showing just the waveforms in the time domain. And the spectrum on the bottom shows 10 hertz and 11 hertz. There is no 1 hertz present, even though there's a temporal interference. But the frequency content really is only at 10 and 11. So that's the one example using the Boyden deep stimulation. The other example was using an amplitude modulated 10 hertz in this case. You see the 10 hertz carrier that is amplitude modulated at 1 hertz in this case. And again, the spectrum doesn't include any 1 hertz. It includes 9 hertz, the difference, and the sum frequencies. So it includes 9 hertz and 11 hertz. So it's interesting that the brain thinks there's a frequency present when it's not really present, and it required that low frequency modulation, even though it's not part of the signal itself. So TDCS alters membrane potentials and alters the probability of a membrane allowing a firing condition, whereas TACS, and so that can have a relatively long range interaction in time, whereas TACS demonstrates a interaction between synchronous activity, and that can happen on a shorter time scale, which might be interesting for modulating and looking at the effect of the stimulation on an ongoing task. So the problem with all of this is that if you look in the literature, there's a huge amount of inconsistency. And so a number of studies have pointed out the inconsistencies. That is, one study might show an enhancement of a particular effect when applying anodal TDCS. Another might show exactly the opposite. And so a number of studies have looked at this. And we think that possibly the interaction between the inconsistencies is the result of targeting that may not be perfect. And it might be hard if you put montage of electrodes, for example, on different parts of the brain. They might, a small change in their location might significantly affect the regions of the brain that are stimulated. So we wondered, where is the stimulus region? In TMS, the magnetic field is not really affected very much by changes in local conductivity from CSF and so on. So the magnetic field is pretty well known where it goes. However, what you have to do is calculate the electric field, because it's not the magnetic field that does anything to the brain. It's the electric field that makes a difference, and that will depend on the electrical properties of the material, and whatever currents are generated that make any difference will depend. So even though the magnetic field is pretty well known and can be calculated easily, the electric field and where it goes is not necessarily. Similarly with TDCS, especially with large electrodes like people tend to use, although we use much smaller ones, where does the electric field and the current really go? That's an open question. So what we plan to do is, so a lot of people have looked at using modeling with various approaches and various montages to look at where the electric field and where the currents really go when you put electrodes on in various montages, such as this study shows. And some people use high-density EEG to help infer where might the electric field go and where do the currents go. But in any case, you assume that the current is known where you injected it because you assume it's going in where your electrodes are, and you assume some kind of a model for the complex conductivity of the brain, and then you apply some kind of inverse solution to figure out where the current actually is going. But, and you might use some external constraints to help you in that modeling process. But often the models are rather simplistic and do not really provide a very high density look at the brain mechanisms. And instead, we think that maybe you should just measure it. And you can measure it by the fact that you're doing MRI. So let's switch to looking a little bit at mapping the stimuli in the brain. So MR signals are complex, and usually we throw away the phase information because it's not very interesting when we're looking at typical fMRI kinds of experiments, and because the phase often changes for reasons that we don't know or care about. But in this case, we're going to take advantage of the use of the complexity of MR, and we note that the phase reflects an off resonance. And usually that's annoying to us, but in this case we're going to do it with and without the stimulus applied and subtract them, so the spurious phase will go away. And what we're left with is a change in phase that relates to changes in the local resonance frequency of the spins. And the resonance frequency, as Larry mentioned, is a function of the magnetic field. So if you're putting a current in, you're going to change the magnetic field, and maybe we can measure that. So the field perturbation is going to be pretty small by injecting, say, 1 MA. The question is, can we observe that small magnetic field? And it turns out that if you can measure like a 1 degree phase shift, you have an echo time of typically 30, say, milliseconds. You can look at nanotesla kind of magnetic field changes. And if you just do a back-of-the-envelope calculation of a wire with 1 MA through it, centimeter away, you have magnetic field changes that are on the order of 3 or 4 or 5 times as large. So it seems feasible that one could look at phase shifts in the MR signal and use them to infer the magnetic field. So the mapping approach is to acquire phase images with and without the stimulus applied, and then look at the phase difference and calculate the magnetic field from that. And from the magnetic field, at least, you then know where to look for current changes. So by using Maxwell's equations. So with a TMS coil, you typically have a stimulator that is applied to the coil, and the stimulation is generating a pulse of 4,000 or 5,000 amps for a few hundred microseconds. And what we do is disconnect the stimulator and instead connect a small 1 or several MA DC or maybe AC current source, and then we look for changes in the phase of the MR signal that are related to that change in the magnetic field. And this shows, for example, a figure 8 coil on a subject with a positive and negative going change in magnetic field. That's localized in this case pretty close to the coil, and that's in fact what one sees. So you can look at doing this with TDCS, and here's an example on a phantom. You can see in the upper left where the electrodes were placed with a conductive surface on the phantom, and on the right we're showing changes in the phase shift that's observed with the magnetic stimulation generated, or electric stimulation generated with a square wave modulation in the right. And if you put a region of interest in the phantom somewhere, you can see those phase shifts in the phase of the MR signal, and then you can use those phase shifts to derive the change in magnetic field. And so here's an example with the phase and magnitude activation, if you will, using a sinusoidal perturbation with electrodes over the left and right motor cortex. And demonstrating very nicely the change in positive and negative going change in magnetic field. And so with electrodes over the prefrontal cortex, again, you get a different series of images. Again, it is very specific to where the montage is. And in fact, if you change the montage, you'll get different looking kinds of signals. Now, it turns out that a small change in the montage can make a big difference. So here's an example with F3, F4 montage. And if you move one of the electrodes to super marginal, you see a fairly large difference in the magnetic fields that are changed. So we think that possibly this small change in the montage can explain some of the variability in results that one gets. Now, it's very interesting. If you look at the phase images and you look at the magnitude images from a typical example, we see apparent activity in both the magnitude and the phase. We see in the CSF where the red or the yellow arrows are showing. Is there a field in the CSF that's being induced by the current, in this case with DLPFC electrodes? Well, we don't expect to see a magnitude change. We don't expect to see a change in signal amplitude from a phase, a small B field. So what's going on? Why are we seeing an apparent change in phase shift and amplitude shift in a region of the brain that's very localized to CSF? Well, it turns out that you can not only have a change in the phase from a change in the frequency of the spins, but also if they're moving. And we see that the phase in a first order motion due to velocity will give a phase shift also. And if the phase shift is large enough, that it could also reduce the amplitude through intervoxel dephasing. So here is that amplitude map again, and there's a T-score map just showing very large positive and negative changes in the amplitude of the MR signal related to the sinusoidal perturbation. And if we look at a movie, and if you could start the thing on the right, please. Because I can't do it. If you just click on it. Well. Yeah. So if you look carefully, the thing looks like spins are moving up and down in the ventricles. So there's a tissue or some kind of a fluid motion that is very, very well localized to this region of interest, and it is perfectly in sync with the 50-second time constant of the sine wave. And so if you look at where this is occurring in the brain, it's in the ventricles. The little yellow arrows on the bottom are showing where the electrodes are over DLPFC. So it's very interesting that apparently we can change physical properties of the brain by putting an electric current in. And maybe that has something to do with the lymphatic system and pumping of CSF and so on. But it's very interesting, then we have to kind of think about this carefully because it's possible that we may be making physical changes to the brain by putting TDCS, or in this case, slowly varying TDCS signals in the brain. Now, the phase only yields the BZ component. We can only affect and make changes or make measurements in the Z direction of the magnetic field, and so we can't get a complete picture of the current. Single measurement would not allow us to totally derive the current. Nevertheless, if you look on the bottom, the green lines show the induced the current that's coming just from those components that we can measure, and it may give us some information, but it isn't a complete set of information just by making one set of measurements. So a possibility is to put the person in and do 3 measurements with the brain moving to the left or right, or tilting back and forth. And by doing so, you'll get a component of the magnetic field shift just from the axial images. We made a phantom containing a current loop in a jug of water, and we could put current through that little Tygon tubing. And here's the magnitude images just showing that, in fact, it was too thick to fit in one slice, but by making the measurements in 3 different orientations, we can make a map of the field. So in summary, transcranial electric or magnetic stimulation can make changes that reflect a causal relationship between what you're doing to localized regions of the brain and nodes of resting state networks, or one can look at the change that happens during a task. And TDCS alters the electric environment of the spins of the neurons, and therefore can modulate their ability to fire or to be inhibited, whereas TACS alters the neural oscillatory relationships. Variability is quite rampant in the literature. So if you look across the literature, you'll find very large changes in the conclusions that people draw from doing ostensibly the same experiment. And we think that maybe that's because of errors in targeting and the fact that both magnetic and electric stimulation are not necessarily very focal, although the Boyden group showed that possibly by using temporal interference, one can make a much more focal region of activation, and that's turning out to be possibly very interesting. But making measurements would be useful, and induced changes in the field may be observable with phase contrast MRI. Mapping then can be used to draw causal inferences about changes in the brain, and that can be very useful. So it may reflect different relationships that you might not even understand when you put the electrodes on, where the currents go. And also we're finding that it's possible to make modulation of tissue structures within the brain using electric stimulation. And that's interesting from both the standpoint of safety and other things, but also it may tell you some things we may be able to understand about the brain. So with that, I will stop.